Here is a step-by-step procedure to solve a genetics problem. This is for students of Bio 196 of Bishop's University. So the first step when trying to resolve a genetics problem is to identify the problem exactly. So what is it exactly? Is it a monohybrid or a dihybrid cross? So this is the first question that you should ask yourself. So first read the problem and determine whether the problem looks at one trait, for example the color of the fur, with two versions. So for example spotted versus non-spotted, or a certain color versus a different color. And this is a monohybrid cross because we look at, in fact, one trait, which is the color, and this trait has two different versions. So in the example we give here, in the little problem shown here, we can see that it is a monohybrid cross. If, in contrast, the problem looks at two different traits, so for example, the color of the body and the length of the wings, for example, with each of the trait having two different versions, so let's say the body color can be gray or ebony, or and the length of the wing can be normal or vestigial, then you're looking at two different traits with each two possible versions. So this is a dihybrid cross. Another example here is given where we have normally pigmented people versus albinos, and PTC tasters versus non-tasters. So this is another example of a dihybrid cross. If you're not sure how to determine this, we will look at this a bit more precisely. So here you read the first problem, and you see that we are talking about the spotting of the coat in Holstein cattle. So if you look at this, the trait is the color of the coat, and it has a version that is spotted versus a version that is non-spotted or solid. So this is one allele that encodes for the spotting of the coat, and one different allele of the same gene encodes for a solid color pattern. So again, it is one single trait with two versions, and it is therefore a monohybrid cross. If you look at the problem down, then you look at the question here. So we say that 70% of American people get a bitter taste from a certain drug, the PTC, while others do not. So the first trait we look at is the capacity to taste PTC. And the second trait that we are looking at is whether people are normally pigmented or if they are albino, so they have a total absence of pigmentation in their skin. So this is the second trait, the color of the skin, basically the presence of a pigment in the skin or not. So you have for each of these traits two versions. In the case of the capacity to taste PTC, the gene has two different alleles. One encodes for the capacity, so in fact the presence of a receptor, while the other version or the other allele doesn't encode a functional receptor, so it is a condition where you cannot taste the PTC drug. In the case of the trait number 2, so the gene that encodes that is responsible for the color of the skin, the pigmentation of the skin, there is one version where the pigment is present, so the skin is normally pigmented, or a version where the pigment is not present and therefore the person is albinos. So in this case, we look at two different traits, therefore it is a dihybrid cross. So for the remaining part of this video, we'll concentrate on a monohybrid cross, so the problem with the spotting of the coat in the Holstein cattle. So the next step then, the step number 2, is to identify the dominant and recessive alleles, the genotypes and phenotypes of individuals that are presented in the problem. So if you look at the problem here and you take the time to read it, you will see that here it is said that the spotting of the coat is due to a recessive gene, while the solid colored pattern is dominant. So you will use the first letter of the trait, so solid, for example. And if the solid allele is dominant, then you put this letter in capital, so you will use a capital S to show the solid allele. At the same time, you will qualify the recessive allele. So the code or the convention for the recessive allele is to use the same letter as for the dominant trait, but we use it this time in lowercase. So here, anyway, it is the same letter because spotted and solid have the same letter. But if it was a different letter, you would use the letter of the dominant, but put it in lowercase. So this is the convention, and you always have to use the convention by geneticists to work with your problems. So now let's qualify the genotypes and the phenotypes. So first of all, the spotted phenotype that you can put in bracket with a little s, so it shows the recessiveness of this phenotype. So the recessive spotted phenotype is due to a homozygous genotype. So it is because it is recessive, so you need to have 2 copies of the recessive gene. So the genotype is shown with the 2 letters, so the 2s in lowercase. If you look at the solid pattern, then the phenotype is indicated in brackets with a big S or a capital S. And this can result of both a homozygous or a heterozygous genotype because the S or the solid allele is dominant. So the genotype of these individuals will be big S small s or big S big S. Now, it may happen that in a problem, you're not told which one is dominant and which one is recessive. So in this case, you will have to determine that yourself. And an easy way to do that is to look at the first generation or the F1 generation, and they usually express the dominant phenotype, so it will tell you which ones of these alleles is dominant. If it can happen sometimes that the F1 express a third phenotype that is different from both parents, so which in this case would be non-solid and non-spotted. And in this case, it is a particular condition that is a case of codominance or incomplete dominance, and we will see that in different kinds of problems. So if we go on with this problem, we will solve the number B item, so the fact that here you're asked to determine the results of a cross between a spotted cow and a homozygous non-spotted cow, so solid colored cow. So we will do this cross, monohybrid cross, and determine the phenotype, the genotype, and the alleles produced at each generation up to the F2 generation. So the first step is to write the cross and all the known data. So what we know of this cross, first of all, is the parents. So their phenotype is spotted and solid. Okay, so with the spotted indicated in a little s in brackets and solid in a big S in brackets. Their genotype is double little s for the spotted because it is recessive, so forcefully it will be a homozygous recessive genotype. For the solid pattern cow, we know it is said in the problem that it is a homozygous cow, so it is forcefully 2 big S that go here. Now, what are the possible gametes generated by these parents? So this is the step 4. You have to determine the possible gametes of the parents. So the possible gametes will result from meiosis, so the segregation of the 2 different genes or the 2 chromosomes that carry the different genes in the parents. So instead of the double little s, you will segregate these 2 in 2 different cells or 2 different gametes. So after meiosis in the spotted cow, you would have gametes with little s or little s. So basically, all the gametes will be carrying a recessive spotted allele. Same thing for the solid colored cow. So you will have after meiosis the segregation of the two genes, but because they are the same, because the cow was homozygous, then all the gametes will have the big S. So the gametes produced by these parents are 100% small s for the spotted cow and 100% big S for the solid cow. So the next step now will be to establish the genotype and the phenotype of the F1. So starting from the gametes that we have, we know that all the individuals of the F1 generation will be coming from the fusion of a gamete of the first cow and a gamete of the second cow. So every individual of the F1 will inherit one gamete from each parent. So the spotted parent can only give little s, and the solid parent can only give big S. So as a result, the genotype of the F1 will be 100% carrying one big S and one small s, because every cell of these F1 individuals will receive half their genetic content from a first parent gamete and the second half of their genetic content from the second parent's gamete. So in terms of phenotype, 100% of the F1 will be solid, so you can indicate that in brackets with a big S. Now the next step, or step 6, will be to determine the possible gametes produced by the F1, because we need to go to the F2 generation. So the F2 generation comes from mating of F1 individuals together. So our F1 individuals, we know, we have just established that they are 100% solid in terms of phenotype. So you can indicate them in brackets with a big S. So they are now the two parents, okay, even if we call them F1. And we know that their genotype is big S, small s in all cases. 100% of the F1 are heterozygous, so they have one solid allele and one spotted allele. Now, the same thing will happen. They will produce gametes. So, with meiosis, the gametes will arrive. So, you will segregate the two different versions of these genes, the two different chromosomes, in fact. So, you will produce gametes with big S or gametes with small s in all of these individuals. So, the two parents will produce 50% of spotted gametes and 50% of gametes carrying the solid allele. Now, once you have this, you need to determine the genotype and the phenotype of the F2 individuals. So you remember that the F2 individuals will inherit one gamete from each F1 parent. So because the F1 has equal chances to contribute a small s or a big S, then what will happen in the F2 will be several possible genotypes, because all the combinations will be possible. Either you will have the two gametes carrying the solid allele that will go together, so it will create a big S genotype. In fact, if you count the chains to obtain this genotype, you just have to remember that in order to obtain this genotype, you need that one parent gives a big S, the chance is 50%, and that the second parent also gives a big S, the chance is again 50%, because the first condition and the second conditions are needed to obtain this genotype. You need to multiply these two probabilities, so it is 0.5 times 0.5. It gives you a probability of 0.25, or 25% of chances to obtain this genotype in the F2 offspring. Now you can repeat the same thing with all the other possibilities. So this same gamete of the first parent can go with a second or the different gamete of the second parent. So it would give you a genotype big S, small s in 25% of the cases. Now, the second gamete of the first parent can go with the second gamete of the second parent, so 25% of chances to obtain two small s. Or last possibility, the second gamete of the first parent and the first gamete of the second parent meet and produce a F2 offspring that carries a small s and a big S, so it gives you 25% of such offspring. Now, if you notice, here we have two different conditions that give you a heterozygous genotype. So what you can do here, you can have a heterozygous genotype that is obtained either in the fact that the first parent contributes a big S and the second parent contributes a small s, so it gives you 25% of chances, or if it is the second parent that contributes the small s and the first parent that contributes the big S. So it gives you a second possibility to obtain the same thing. So when one condition or the other will do the same thing, you can add the two probabilities, so it gives you 25% plus 25%, so 50% of obtaining this. Now, if we look at the phenotypes of these two individuals, so the possible phenotypes obtained. So if we look at what we have less, we have only 25% of individuals that will present the two recessive alleles. So these individuals will have the spotted phenotype because they are homozygous for this recessive trait. So it will be 25% of all F2 individuals that will be like that. However, all the ones that carry at least one big S allele or one allele encoding for the solid pattern will show the solid pattern trait. So it will give you 75% of solid colored cows in the F2 individuals, and these result from the addition of the homozygous solid and the heterozygous solid and spotted individuals. So in total, 75%. Now, it is possible that in some problems you're not asked for the probability or the percentage, but you're asked rather for the ratio of individuals obtained. So in this case, you should answer under a slightly different format, with the genotypes in F2 being indicated like big S, big S, big S, small s, and small s with 1/2/1, and the expected phenotypes being solid over spotted or 3/1. This is the way we indicate the ratio in contrast to the probabilities. You may rather use a Punnett square to find the F2, so it is a second possibility, a choice that you can make. So in this example, we will do the same thing. We have the gametes that are produced that are the same as what we indicated previously, with each individual in F1 generation creating big S or small s alleles. You can put these alleles in a Punnett square. Like one parent goes on one side, the second parent goes on the other side of the square, and then when you combine all of these possible gametes, like a big S with a small s gives you 25%, etc. So you obtain the exact same result. So possible genotypes, big S 25%, big S small s 50%, and small s 25%. In terms of phenotypes, same thing, 75% solid, 25% spotted. And again, if you are asked for ratios, you should rather write them in terms of 1/2/1 in genotypes and 3/1 in phenotypes. Now, the last thing you have to do is to double check. So first of all, what were you asked to do? If you were asked to identify the possible offspring, you just need to cite the results in terms of qualitative results. So show the phenotypes and the genotypes. You don't need to give numbers. If in contrast you are asked to calculate the ratio, then you should give a ratio like we just said, and this one it is quantitative. If you are asked to calculate the probability, then you need to give quantitative results again, so numbers, but you give them in the form of a percentage or a fraction. Next thing, are all your calculations correct? In fact, if you add all the probabilities or the percentage for each individuals in each generation, you should add up, each generation in fact should add up to 1 or 100%. This will make sure that your calculations are correct. Thank you.